Hello everyone, I'm Maria and I'll be talking about the research I did in Polshio's lab this year. I was lucky enough to do my honors project on a topic that I'm very passionate about, extension of healthy life by killing of aged cells. I'm interested in this because I think it can be a potential solution to a problem that we're gonna face very soon. The growing population worldwide is going to result in increased group of people over 65, and by the year of 2060, people over 65 in the EU would outnumber the people under 15 by 3 times. The working group people from 15 to 64 would also decrease. And this is not just EU data, it's a worldwide problem. We see that population over 85 would triple and even quadruple in some countries where the situation is more severe, such as Asia. So where is this problem coming from? We see over the past couple of decades that lifespan has been dramatically increased, but there is no corresponding extension of health span or the period of a person's life that is spent in good health. Here we see that an average lifespan will be around 85 years now, but a substantial amount of that time is in red, and red signifies disease. After the age of 50 to 60, we start getting increased risk of getting one of those age-related diseases. And so what we want to do is increase the health span of a person, which would result in extension of lifespan. But our main focus is to increase the quality of life of billions of people rather than make them live forever. So what is aging? It is the accumulation of damage in an organism as it progresses through life. And as this damage becomes too much, a lot of diseases start to arise. So the aging is the underlying cause of all those diseases. It has characteristics such chronic inflammation and tissue degeneration, which are common with all the age-related diseases. So what we want to do is, rather than treat all of them individually, just treat the underlying cause, which is aging, which would postpone all of the diseases as well. So something I need to make clear is that chronological age, or how many years we live, is not the same as biological age, or the state of the body and the cells. Allow me to illustrate. This is Russell Crowe when he was 4 years old. And that's him 10 years later. You can clearly see the aging process. This is Keanu Reeves, he hasn't changed at all. And this goes way back, a lot of people have noticed it. You can see on keanuisimmortal.com. There are some suggestions that he's a vampire, but I'll get back to that later. Now my main point here is that people age differently. Age at a different rate, but it happens to all of us, and that's dependent on genetic changes or environmental factors, lifestyle choices, etc. But we also need some direct therapies if we want to significantly slow down aging. So we have some of those available today. One of the most studied one is dietary restriction. Researchers have shown that when we decrease the calorie intake of an animal, we can see lifespan increase of 50%. But how many of you would like to starve their whole life? Right, that's not. It's not the plan in humans. Another therapy, speaking about vampires, blood transfusions. Blood is taken from young people and put into old people, and that's shown not to just slow down aging but also reverse it, and it's actually done in humans. There is a company in the US called Ambrosia, does that. There are also some drugs that are currently under development, or this one is already been on the market for a long time. Most of them work in a similar fashion as diet restriction. They mimic the effects of diet restriction without actual diet. Metformin is a type 2 diabetes drug, which have also shown anti-aging effects, but it is not proven as an anti-aging drug yet, so it's not used. And there are a number of natural compounds, such as resveratrol. It's found in red wine. However, its concentration is not nearly enough to slow down your aging process, even if you drink a lot of it. So we see that all of those potential therapies have some drawbacks. That's why we're looking at a different strategy. We want to remove senescent cells, and senescent cells are a certain type of aged cell, or also called zombie cells, because even though they're alive, they're not quite like the normal cells. They don't divide anymore, they don't produce new cells. They have undergone a transformation, which is due to stress or damage. And this transformation causes them to release molecules that are damaging to their surrounding cells, and it also makes them immortal. Now, usually healthy cells, when they're stressed or damaged, they would essentially kill themselves or sacrifice themselves so they can protect the whole organism. However, senescent cells don't do that. That's why as people age, there is more and more senescent cells in their tissues, which results in tissue degeneration and inflammation. Here are some healthy cells in the first panel, and after damage or stress, some of them turn senescent. They start releasing a lot of those damaging molecules. This results in some of their neighbor cells turning senescent, and usually in the young healthy organisms, those cells will be targeted by the immune system and destroyed. However, when we age, the immune system gets weaker and weaker, and it cannot destroy the cells anymore. That's why we need drugs. So there are some drugs that have been researched, and they have been shown that when they remove senescent cells, there is tissue regeneration and restoration of physiological function. However, our strategy is a bit different. In our lab, we use microvesicles, and microvesicles are a type of extracellular vesicles, which is basically a tiny package of cell which buds off the cell wall, and it contains different molecules which have been shown to have regenerative effects and to remove senescent cells from animal models. So how might the microvesicles work as senolytics, or senolytics are drugs which remove senescent cells in animals? So far we know that the senescent cells don't die on their own. We know that when we remove them, there is restoration of physiological function, and we know that microvesicles allow for tissue regeneration. So when we put that all together, we're asking, do microvesicles actually kill the senescent cells? And this is where my project comes in. Here is what I did. I culture cells under different conditions. Some of them are stressed. We wounded the plates. We physically scratched the cells to induce damage. And then we treated some of them with microvesicles. Now, when cells are under different conditions, they start activating different genes. So we isolated the RNA from the cells, which is the copies of those genes, and we measured their levels to see if we have more or less of those RNAs. And after analysis, we got this. This graph would show us whether microvesicle treatment promotes cell death in senescent cells by activating suicide genes. What we see here is gene levels. There are three groups. The first one is the untreated control, or normal healthy cells. The gene levels of those cells, which are all associated with senescence and cell death, are usually in those amounts in a healthy cell. In the second panel, we see that they're massively dysregulated as there is increase in senescent genes, which are the first blue ones. And then in the third panel is after treatment. And what I want you to take from here is just that there is a clear difference in the pattern. I know it's a bit overwhelming. That's why I've extracted the data from the previous graph here, where we see the individual genes with biggest change in expression or in activation. Now we see that some of the genes go up and some of them go down, but as a whole we can summarize that they act to promote cell death. This big yellow bar is a gene that promotes cell death, and all of the other ones that goes down actually stop cell death from happening, so it's good that they go down. And from all of this data, we can conclude that our preliminary analysis shows that microvesicles can induce cell death in senescent cells. But as I mentioned, this is only preliminary data, and we need further experimentation to actually prove this. However, if this is correct, it will be a potential therapeutic to remove senescent cells from tissues and allow for tissue regeneration and reduction of inflammation, which would delay the onset of age-related diseases. Thank you. Any questions?